Hello, everyone, and welcome to our webinar today. Before we get started, I just wanted to introduce myself. My name is Alex Corrali, the U.S. Field Application Scientist here at AYOXXA. And today I just want to give you a brief presentation and a short introduction on our LUNARIS Multiplex platform, along with several use case applications from our customers and collaborators for aiding your research in cell therapy. As a housekeeping note, please feel free to utilize the chat function box to post any questions you may have. And at the end of the presentation, I will try and get to them. If time runs out for whatever reason, or we can't get to your questions, one of us from our team will gladly respond via email. Thank you again for taking your time out of your schedule to attend, and let's get started. So most of you today were invited due to the simple fact that your research of interest is focused on cell therapy and/or immuno-oncology. Now, I won't go into too much detail or a background of cell therapy, but rather just touch on the point that cell therapy, as we know, is a relatively new treatment option introduced to the market in 2017 with Kymriah, as well as Yescarta, to just name a few. And this treatment option's really taken off with more than 1,000 compounds now in clinical development and testing. As you may know, there are certain associated risks with off-the-shelf cell therapy, such as systemic rejection, as well as cytokine release syndrome, which is a hallmark adverse effect for Kymriah, as well as neurological disorders as seen with Yescarta. There are several approaches that one could utilize to minimize these risks, for example, correlating immune profiles with clinical outcomes, as well as identifying longitudinal cytokine, chemokine, and angiogenic factors involved. And that is where LUNARIS can play an essential part. Our LUNARIS technology can better help your research needs for cell therapy by easily identifying and quantifying cytokines, chemokines, and growth factors of interest in various sample matrices, identifying biomarker signatures that correlate with disease identification and/or progression that could be used as a diagnostic tool for your research, and efficient and economic testing. Our technology is sensitive, robust, and precise. Furthermore, our technology can allow you to gain more from less, using as little as 3 microliters per well for testing. This can help you champion the 3 R's of research, replacement, reduction, and refinement when working with animal models. For those that don't know, multiplexing is a tool that allows its users to identify and quantitate multiple biomarkers or analytes of interest at one time, rather than performing individual experiments or assays. Essentially, you are running multiple sandwich ELISAs in one well. Our LUNARIS technology operates on a planar bead-based detection system for multiplexing. Pictured here in the middle is an image of our biochip, which is the heart of our platform. These biochips come in a 96-well format, and in each well, there's approximately 10,000 microcavities that houses our proprietary beads. Now, what sets us apart from our competitors, and trust me, you will know this by the end of the presentation, is our low volume needed. We only require as little as 3 microliters of sample per well. Now, our platform consists of 3 components: our kits, our reader, and our software. We have over 100 analytes to choose from for both human and mouse models in the application fields of immuno-oncology, inflammation, and ophthalmology. For time's sakes, I won't go over each of these kits that we offer, but I do encourage you to take a look at our website for more information. Now, each of our kits, sensitivity-wise, can measure as little as 1 picogram per milliliter, depending on the analyte, with a dynamic range of 3 to 4 logs. Furthermore, you can see the accuracy and precision from 3 separate experiments from both the within run and the between run percentages on the side of the graphs. And if you take a look at the top 2 graphs, you can see that the CV, or coefficient variance, is below 20%. Now, next up is our reader, which is a dedicated benchtop device. It's about the size of a laser printer. It's a truly plug-and-play system. Once you finish the assay portion, all you have to do is place the plate into the reader and press play. One of our biggest advantages is that we do not require any scheduled maintenance or cleaning. We also give you the option for a flexible format. Depending on the model you choose, users either can use 1 biochip for 96 wells, up to 300 or up to 4 biochips or 384 wells. Reading wise, it takes about 10 minutes per biochip. So you're looking anywhere between 10 minutes to 1 hour. And lastly, our software. This program is very easy and intuitive to work with, but the best part, in my opinion, is you are greeted right off the bat with the standard curve results after uploading your experimental files. From there, you have the options to view your results either in the heat map setting or within the standard curve as shown here. Our software also has a built-in feature that will recognize any outliers that you may choose to delete or take off from the experiment, or you may want to leave it on, as well as we give you options of exporting all the raw data in either a PDF or an Excel format. And then one last feature that I would like to mention is that our software allows you to create reports and graphs as shown here for easy record keeping or for those last-minute lab meetings. Now, that was just a brief introduction. If you do have any questions, please feel free to ask. If you'd like to know more about the system, contact one of us. Our email will be displayed at the end. We can give you a more detailed presentation on the technology. Just wanted to do a brief one. And then at this point, we're going to go over about 4 different use cases to show you how our technology is being used in the field from our customers and collaborators. So for our first use case example, we're going to take a look at a group studying graft-versus-host-disease. Graft-versus-host-disease is a common complication in stem cell transplantation that involves leukocytes in the graft being activated by the foreign host tissue, in turn beginning to attack the recipient's body. This can cause uncontrolled inflammation that can lead to multi-organ damage. There has been called for a need for establishing a biomarker signature to guide early intervention. Our partner and collaborator, Vicky Besoides from the Beth Israel Medical Center, aimed to do a risk assessment study for immune reconstitution after cord blood transplantation. In this initial study, 27 patients were included. Blood draws were taken at baseline plus 6 different time points and followed for 24 months. Both serum and cell fractions from white blood cells were collected and profiled using our LUNARIS human 11-plex cytokine kit. Data was then analyzed to determine which biomarkers were up or down regulated for each cellular subtypes at different time points, which I'll show you on the following slide. Overall, the impact that this study has had could show that an early intervention for acute graft-versus-host disease. And just kind of touching up and kind of going further into their data, pictured here again are those correlation levels for each biomarker and cellular subtype. And you could see that there is a biomarker signature apparent within 1 to 2 months. They found that inflammatory cytokines, interferon gamma, IL-6, and TNF-alpha were increased 1 to 2 months after transplant, followed by a subsequent decline with the exception of interferon gamma, which remained relatively stable throughout the whole study. In addition, they found that IL-6 levels during the post-transplant period positively correlated with the development of graft-versus-host disease. Again, this group's impact may lead to early detection and intervention for graft-versus-host disease and has led to a follow-up multi-center, multi-participant study, which is currently going on. And for our third use case, we'll be taking a look at CAR T cell therapy in prevention and therapy development. Now, CAR T cell therapy is a pretty popular topic these days. It is being studied as an effective treatment for various types of cancers. Here, isolated T cells from a patient or a healthy donor are genetically engineered to express the chimeric antigen receptor, or CAR, that targets tumor cells. So essentially, it's a living drug against cancer cells. However, safety and efficacy must be assessed in preclinical studies. One of the main toxicities involved in CAR T cell therapy is cytokine release syndrome or cytokine storm. If we take a closer look, increasing concentrations of interferon gamma and TNF alpha are caused by CAR T cells and tumor cell lysis. Activation of bystander immune cells and non-immune cells, for example, endothelial cells. Stimulation and expansion of monocytes and macrophages. Macrophages produce high concentrations of inflammatory mediators, particularly IL-6, which then enforces the clinical presentation of cytokine release syndrome. Unfortunately, for those who are watching on our YouTube channel, the primary investigator has requested to omit the following data as the study is still ongoing and yet to be publicly available. I would like to mention that cytokine measurement is an important tool for early intervention, especially for those receiving CAR T cell therapy. And that brings me to another study. Whether you're testing serum or plasma, LUNARIS allows you to achieve single animal kinetics due to this low volume requirement. Now, this study was done from a academic research partner out of Germany, where they used both wild type and knockout mice, infected them with a specific bacterium, and serum was collected at different time points within 24 hours. Now, traditionally, multiple blood draws on mice can be tricky for such short time period, especially for the animal's well-being, and therefore many groups pool samples to achieve volume requirements. But pooled samples can also increase inter-animal variability and can leave you guessing what may have happened, as shown here for both the mean graphs from GM-CSF as well as IL-13. But by conducting the single animal kinetics, now you have the option to looking at each individual animal and see which animal may have caused an increase or decrease, or just kind of that's not within the norm, and then you can go ahead and further analyze your results. And now our last use case, but certainly not least, with LUNARIS, you can also measure markers from cell culture supernatants. In an application note published by us in 2019, one of our associates showed that cytokine profiling was achievable when culturing three-dimensional cells using a Biomed or a AIM DAX1 microfluidic 3D cell culture chip. Excuse me. Briefly, hep G2 and monocytes were prepared in a collagen-type solution and injected into a gel channel. Once polymerized, T cells, media, and a live dead cell marker was injected into the media channel for culturing the 3D structure. High content imaging was then used to determine live dead cell ratio, and the supernatants were collected for profiling using our human 11-plex cytokine kit. Now, when looking at the results, you can see the presence of CD4, with presence of CD4, CD14 positive monocytes, IL-8 levels were increased in all groups. Expressions of IL-5 and IL-6 were increased where engineered T cells were placed. And then just this quick summary. With LUNARIS, again, you can achieve more from less. Remember, only 3 microliters of sample of volume is needed. It's robust and reproducible using our planar bead-based detection system. We offer a flexible and scalable format, anywhere from 96 wells to 384 wells for high throughput screening. And all our kits are validated for specific matrices. Our ophthalmology kits are validated for vitreous and aqueous humor. And from there, again, this is just, I want to just take the time, if you'd like, please check out our website. We have a list of selected publications where the LUNARIS technology has been implemented. And if you have any questions, I'm going to get to them here shortly. I do thank you for your attention. If you have any further questions, please feel free to email anyone from our team at sales@ayoxxa.com. Thank you.